much, Brad, and thank you, Genome BC, for organizing this event. I think it is very important to have these meetings regularly in getting the BC genomics community together to foster closer relations and as well as to initiate new collaborations. Today, what I'm going to do is give you a brief introduction to our new project from Genome Canada, Genome BC, in the last bioinformatics computational biology competition. It is called Next Generation Bioinformatics for Clinical Genomics. And I would like to also share with you some past success stories that were partly enabled by funding from Genome BC, and actually those are the types of small funding that enable this large-scale project. So it is going to be about using de novo assembly in personalized medicine. And the rough overview of the project is it is going to have three specific aims, three pillars, if you will. We're going to be working on the analysis accuracy, the turnaround time, and clinical reporting. So those have totally new meanings when you apply a research project into the clinical domain. And I will have only time to go into giving you some flavor about the analysis accuracy today. So when we talk about translating a bioinformatics tool into the clinical domain, any experimental system, and by experimental system I mean anything, everything that generates data for analysis, is operating somewhere on a receiver-operator characteristic plane. Okay? So it's a concept that is borrowed from a statistical control theory, and what it is it is putting your operation, it is putting your analysis system to a dot on a plane where one of the axes is typically sensitivity, the other one is one-minus specificity. So what you would like to do is, of course, to be on the upper left corner there. Okay. So, okay, we are translating this into the clinical domain, and then when we're discussing this with our clinical collaborators, and of course from a research perspective, when we start talking about false positives, false negatives, they look back at us as if we are from Mars or from Venus, if you're related. So they don't want to hear it. They want to operate at that corner. And what do we say? Sure, right? So we want to push our analysis over to that corner there. In bioinformatics, when you are analyzing next-generation sequencing data in projects like clinical genomics projects, you are employing one of the two paradigms. You either assemble your data first, assemble your reads first, then align them to align your resulting context to a reference genome to do your downstream analysis, or you align your reads first to a reference genome and align your reads in that reference genome using the references to your guide to do your downstream analysis. Now, what we're arguing here is if you are using an alignment first approach, that you are biasing your analysis results, as opposed to the assembly first approach where you let the data tell you what the story is, and then you interpret what that story is. Okay, so in this example, for instance, you are looking at a cassette repeat of a motif of 7 bases, and if you align your reads allowing for mismatches, you are going to miss that insertion. All right, so, and I don't want to say the alignment-based methods are bad or anything. They are good, they are fast, they have their niche. But if you are solely based, alignment-based analysis methods, if your study is based on analysis, on alignment-based methods only, then you are missing things. So what is the assembly problem? The assembly problem in a nutshell is identification of partial and ambiguous read-to-read overlaps and merging them into longer sequences called unitics. And if you have a longer range connectivity information, you bring those unitics together to form your context. And if you have further linkage information, but if you're not sure about the exact sequence in the middle, then you can build them into further longer scaffolds. Now, our group was one of the first to bring de novo assembly technologies to short-read platforms with our tool called this, which was, again, partly funded by Genome BC. It is a popular tool. For example, if you type this to Google, you would see our download page in first page of results. So we win the popularity contest. But what do we do with it? What we do with it is we build downstream analysis tools based on that platform. One of the analysis tools that we built is called Transibis. It is for assembling and analyzing transcriptome sequencing data. And a couple of years back, we published our results. Remember, the top left corner is the good one. That's where you want to be. And in that study, we compared our results to alignment-based methods to show that, yes, we are more sensitive, yes, we are more specific. So that there's some reason why you should be using this tool. Another example is the Barnacle Pipeline that we built, again, based on this technology, to identify partial tandem duplications and internal tandem duplications in RNA-seq data, transcriptome sequencing data. Again, the Barnacle Pipeline is unique in the sense that it is the first, and as far as we know, the only high-throughput technology that would identify these tandem duplications. So the alignment-based methods do have a hard time identifying these. Why is it important? It is important clinically. For example, in the acute myeloid leukemia samples, you would want to know the three IPDs or MLL-PTDs because they are clinical biomarkers. There has been a good community uptake of our tools. To date, we have over 500 citations in the literature. And in the last couple of years, we have been using these tools to analyze the data from large cancer sequencing projects, including the Cancer Genome Atlas, the TCGA project of NIH, International Cancer Genome Consortium, ICGC project, and other large-scale genomics projects. And we're always looking for more collaborations. So of course, these tools are developed by many talented individuals, including Jared Simpson, Sean Jackman, Tony Raymond, Gordon Robertson, Kang Neb, David Chu, and others. But I would like to end my talk today with acknowledging my project team. The tools that we're going to develop for the clinical domain, that we're going to translate into the clinical domain, are going to be developed in my lab at Genome Sciences Center in collaboration with Stephen Jones. And they are going to be applied to the clinical samples at the Center for Clinical Genomics, a CAT-accredited lab at the Genome Sciences